We can see one who is quite very fat in the middle there, and of course others being a little bit slim. Then we can see the tall one, but we can also try to switch to the profession and see. We have a musician, we probably have a witch doctor somewhere. I don't know how to define that one. Of course, but these people are coming from the same grandfather or grandmother. And so the question to ask ourselves is, how does this come about? And genetic diversity emerges in humans from about 4 fronts. Number one, at the time of the formation of gametes, when the sister chromatids align together during metaphase, there is crossing over and recombination of genetic material. And in terms of gamete formation, every gamete, I like to use the scenario for the women who release 1 egg every month. That egg, the egg that comes out during this month's ovulation, its genetic composition is different from the egg that will come out during the next, largely because of these changes that occur during gamete formation. But that said, of course, we appreciate that all these offspring are coming from the mother somewhere, the father somewhere. So us as human beings who result from sexual reproduction, we are a hybrid of genetic material from the mother and the father. And that's why my first slide actually had this child. So this child has genetic material coming from the mother and genetic material coming from the father. So that alone makes the child different from. So, but you can see why, if this gentleman came from one sperm and the ova from the mother, let's say they are from the same family, his genetic composition is quite different from this one because of the shuffling of genes that happens. So that produces genetic diversity. But at the same time, when we try to understand or disaggregate the impact of genetics on the phenotype and especially behavior, particularly using the twin studies, we get to see that the environment equally has a very big effect in terms of shaping the phenotype. So as a result, even though we carry a lot in our genetics, the environment equally plays a role. So this cartoon depicts a gentleman who is admiring a woman probably at an orchestra because she's playing the violin very well. But little does he know that the genetics that carries this nice talent also probably carries a different undesirable trait with it. So with our genetics comes good traits and of course some traits that we wish to do away with. So as a result of that, we've seen, of course, discussions around gene mutations that are associated with the Parkinson's disease. And there are those we would call rare Mendelian type of Parkinson's disease, where genetic mutation is responsible for the development of Parkinson's disease solely. And if you ask these people why they have Parkinson's, they will be right to answer it's a result of heredity. But on the other hand, we know also, of course, that a lot of the Parkinson's disease results from complex interaction of genetics, environment, and lifestyle. So in terms of personalized medicine, and I thought about two things. The concepts of personalized medicine can be thought of as essentially targeting medicine in such a manner that it respects the genetic composition and the phenotypic uniqueness of an individual. But at the same time, and that's what we call precision medicine, in the sense that you are looking at the underlying genetics and how potentially it could influence outcomes of medication, be it effectiveness, be it susceptibility to side effects, be it the ultimate prognosis. So the cycle for precision medicine starts from diagnosis. And frankly speaking, the diagnosis of monogenetic disorders will take on a different span from the diagnosis of the complex Parkinson's disease. But at the same time, the prediction of the risk in terms of especially the complex Parkinson's comes on in adulthood after 50 years. Is it possible for us to predict this a little bit early? Is it possible for us, for example, to use genetics to predict that somebody could potentially be, genetic testing could potentially be at risk of Parkinson's disease, including, of course, the monogenetic one, especially when we talk about prenatal screening. And then the classification of diseases, knowing which one is monogenetic, which one is not monogenetic, deciding the prognosis, how we respond to therapy or the outcomes of the disease process. How do you target that? What drugs do you use? And this is a question to ask. From the presentations we've had earlier, we've seen that the genes that have been identified for a lot of the monogenetic Parkinson's disease are not present in Africa, or they cannot be replicated in African populations. So the key question you ask yourself is there a different pathway leading to the development of Parkinson's disease in the African population? And then, so in terms of optimizing therapy and potential resistance that could accrue. So similarly, for Parkinson's disease, it is important when we are considering applications in terms of both precision medicine and personalized medicine to look at those things that could produce differences, including the personality and preferences, and we will see how that plays out. The lifestyle, some people have such a lifestyle that they don't have time, or they will easily forget to take their medications. We need to pay attention to some of that. The pharmacoeconomics, affordability, and people have talked about that, how level data is not readily available here. They are giving options for levodopa, Prednisone, of course, makes plant extracts. The comorbidities that people have, and what drugs they are on, which could potentially interact with the drugs that you're putting the patient on. And issues to do with age. So these things one needs to pay very critical attention to. So the concept of, if we move this and start off with diagnosis, and the presentations that have come out, failing to identify or to link the same the synonymous mutations, the non-synonymous mutations with the that are present in some of these genes with the Parkinson's disease in Africa. What implication does that have? And in terms of diagnosis, and what approaches should we be using? It tells us that probably a lot needs to be done in the context of PD, in the context of Africa. Lately, we've been eating a lot of peas, doing a lot of injustice to Mendel's work, but there are complex issues, from all the way from understanding the family history, drawing pedigrees for people who show up with the Parkinson's disease, such that you are able to link, maybe identify the patterns of inheritance. Is it autosomal dominant? Is it recessive? To, of course, the myths that are around Parkinson's. People thinking, okay, this is witchcraft, and possibly opting to go to the witch doctor rather than go to the neurophysician. And of course, the place for the spiritualists in terms of treating these kind of diseases. And these are some of the things that we really need to pay attention to when we are looking at PD in the context of Africa. So this is a very beautiful paper, of course, written by a team of neurophysicists, which is looking at the approach to precision, genomic precision and tailoring of phenotypes and lifestyles to treatment of PD. When we look at personalized medicine, we can think about it in two ways. The precision that is based on the differences in genetics, but also the tailoring that is based on, number one, the disease phenotype, the behavior of the person, and this is where, you know, you move away from the classical approach to the disease, and you deal with the patient seated in front of you as an individual, trying to understand the challenges they have and addressing them in terms of what could be could help improve their outcomes. And then, of course, the issue of having to control for the environment. We talked about, people have talked about taking coffee being a protective factor. Smoking, is that real? The use of sites, and the role for inflammation, particularly in the development of Parkinson's disease, and maybe the use of different immune modulating drugs in terms of preventing the onset and progression. There was a talk about the toxins, the neurotoxins, and the issue of arsenic, exposure to arsenic and magnesium. And these are things that we need to pay attention to when we contemplate personalizing. Now, we can approach personalizing in two ways. The easiest way to approach personalizing is to look at it in context of social groups, all geographic groups. For example, if you look at the context of Sub-Saharan Africa, the fact that we don't have the replication of the genetics presents a unique situation. And then, of course, people have talked about the potential exposure to the neurotoxins. And maybe that should be a point of very key, very major attention in terms of dealing with PD. So, and then if we moved away from, of course, that small picture in terms of, and moved to the comprehensive and holistic personalized approach, the prodromal stage of PD, in terms of genomic medicine, if people are carrying the GBA mutations, you could consider to use histone acetylase inhibitors as well as gene therapy. But we see also from the leucine-rich kinase mutation carriers that you can apply the kinase inhibitors. When we move to the clinical differences in the clinical phenotype or presentation, you know some people will have just the motor symptoms. Others will have the motor symptoms with the non-motor symptoms. And then some probably will be on drugs and will have side effects of these drugs. So as we try to approach these patients, it's important that you look at some of these things and you are able to address them. In terms of targeting monogenic PD, the most important thing is to identify the gene that is involved, and then look at the options that are available. Either you are going to deal with removing the environmental stimulants to reduce the progression to. Many times in our setting, we don't have a lot of genetic screening for diseases happening. But we're trying to build screening programs. And I think that as we go ahead, we will expand the screening programs to include diseases of interest. In Uganda, right now, we are running a screening program for HIV, for sickle cell anemia, and largely because these need to, if they are identified early, something can be done about it. When you go to the US, a lot of metabolic diseases are included in the screening, which we are not doing because largely we can't afford. But moving forward, I think the approach to the treatment of PD in terms of the personalized approach needs to take into consideration some of these things. We can see heat shock protein agonists being used, but I believe that people have talked about vitamin B12 as an antioxidant. I want to also look at the amount of fruits that people take in terms of offering antioxidant and protective effects for people who have monogenetic PD. When it comes to lifestyle, I think largely I've tried to talk about this. It will affect the choice of the drugs that you give, the frequency at which you give the drugs, because like I said, some people might not be able to take drugs more frequently. You probably will have to consider long-term depots. There are issues to do with the side effects and the patterns, the formulations that you use in terms of either they are oral, intravenous, all that will affect how you treat. So this slide, this ideally is the paper that we are referring to that has much of these pictures that I've used is written by Itova, and it's entitled Personalized Medicine in Parkinson's Disease, Time to be Precise. But of course, we look at other things, including the genetic basis. I decided to make my slide and presentation this afternoon, because I realized that when people come from lunch, there is a very heavy likelihood that they will go to sleep. So the best way to do this was to make sure that I have no words on the slides, and there are only pictures. But I have another slide, which in a lecture on personalized medicine given by Eric Green, the former director for the National Human Genome Research Institute. But I will ask you, given what has already been talked about by the previous speakers, should we still go and look at the other slide? Because this was the gist of the matter for my presentation. You still want the other one? Yes. Okay, let's have the other slide. We will run through the other one. But frankly speaking, I try to make my presentations quite easy for people to figure. And Eric Green gave this lecture in 2017, I think. At the time I was doing a fellowship at the Children's National Hospital, Washington, D.C. And it's an interesting lecture, but when you go through it, you're going to find that a lot of things that we've talked about. It's a broad lecture. It's really talking about whether, you know, this afternoon I was speaking with a colleague, and one of the things we were trying to imagine is, we dream big. The concept of personalized medicine is like a dream that we have. But in terms of getting there, it will take us time. But we've seen dreams come to pass. In the 18th century, there was a famous French writer who kept writing fictional books about future technology. Yes, and the most famous book I read when I was in my secondary school is 20,000 Leagues Under the Sea, and he's predicting that at one time people will create this monster that moves under the water. And frankly, at the time I was reading the book, I knew that submarines do exist, and it's impressive that people's future thoughts sometimes can come to pass. So let's just run through this. People have talked about the human genome, total composition, genetic makeup of the human or an organism's DNA. And many times when we use this definition, people get confused. This total makeup of an organism's DNA, it's not that you take the entire organism and no, one cell is enough, it is representative. The DNA in the nucleus of one cell is enough to tell you many things about the entire genome because it's replicated. It's just out of specialization that many things, changes take place. So, 1953, we see James Watson and Francis Crick expound for us the double-stranded structure of DNA, and of course the knowledge that has come subsequently decoding the genetic code, neural experiments, NIH, and then of course moving on to the point where we are able to do the combinant DNA technology, and the early sequencing technologies that come with, of course moving on to the vision to sequence the entire human genome. Took several years. The first drafts being published in 2001, 2003, and there was one supposedly the final one in 2005. But we saw last year a paper come up talking about the non-coding DNA. So essentially, the entire map should be complete. But of course, people have raised concerns too in terms of this is a static reference genome. It does not accommodate the diversity that is present at individual level. Even at race level, does not address that. So when you come to the African genome, things tend to play out differently. I tell my students that if the Caucasian's genome is white light, the African genome is white light, which has been passed through the prism. Essentially, it reveals the entire colors of the spectrum that's present in the white, in white light. In that way, if you looked at the African genome, it's very easy for you to say, okay, purple or yellow is responsible for this disease, which is unlikely when you look at just the white light. And unfortunately, of course, until we had the drive by the Accra Declaration to set up the H3 Africa Consortium to address the gap that was present in terms of the genomic research available, particularly trying to look at how genetic variability influences disease susceptibility. There was underrepresentation of the African genome, but the H3 Africa Consortium, which closed, I think, last year, having run for about 10 years, brought out so many things, but we are continuing to learn many things, including from projects that are trying to look at 1,000 genomes, 100,000 genomes, 10,000 genomes, and helping us to understand how variability could potentially influence. So the movement towards personalized medicine, an emerging medical discipline that involves using genomic information about an individual as part of their clinical care in terms of diagnosis or the therapies that you make. And we take it further and try to say, okay, not just the genetics, but there is also the phenotype. Patients will present with different clinical features and, of course, different challenges. So you need to be able to address that to tailor the medication very clearly to the patient. So the movement towards the realization of genomic medicine, we have a dream. Like June what? June, we have a dream that we can achieve personalized medicine. But the dream is far, and we're moving small steps. We see the issue of the cytochrome P50 and P40 and P26 enzymes in the liver and how they affect drug metabolism. Of course, being addressed in the context of the African population. Slowly by slowly, I think we will get there. I have argued that many of the bad outcomes in terms of cancer treatment come because we are doing very little in terms of understanding the differences, be they the genetic differences of the tumor as well as the phenotypic differences. Breast cancer alone, it's important that you understand what receptor breast cancer is expressing, because that essentially determines the therapy that will benefit the patient. If you don't do that and broadly just give the medicine is not relevant to that patient. So many things that we have to look at. My country, where I come from, we have how many tribes? 50 something? Yes, 56 tribes. And all these come from, they have a heritage. So the Bantu are there, but you could conglomerate the Bantu and say, okay, they come probably from the same, but the dialects changed. We have the Nile Hamites, we have, and one of the things I've thought about in terms of approaching personalized medicine in this heavily diverse tribal country is to contemplate doing a Uganda genome project where we try to map out the heritage patterns and the shared genes across the groupings. So you will find that in the Bantu group, you will probably have 20 tribes, but they share words. So the Bantu from Uganda literally share words with the Bantu from Botswana, from South Africa, and Professor will probably, he's both Ugandan and South African, Nigerian, actually not both, so he probably gets to figure that. But you can see the diversity, and that's what we need to be able to address and how potentially possibly it influences disease outcomes. So frankly, moving forward, the National Human Genome has a strategic plan in this direction. And of course, this strategy is shared across, but the issue has been, the first thing is making sure that the technologies are available. I'm impressed by the presentation showing that the sequencing is coming down to $10 with the Ultima technology. That is close to affordable because frankly, I'm a clinical geneticist, but even simple tests for children born with genetic abnormalities, they can't afford them, and that becomes an issue. So the first thing is to make sure that the technology is affordable. We are able to dissect and identify these uniquenesses at the genetic level. The phenotypic level really is for the clinician to be able to differentiate the differences in how disease is presenting. But the other thing, of course, is the molecules. It will take us several years. There's work being done, say, towards this direction to make sure that technologies become more affordable. Until recently, I think this was the cheapest platform, but it's of course becoming obsolete. In this, you can use it in the field. You see, it's a plug and play. You attach it onto your laptop, throw in your DNA, and you are ready to run. So people are making sure that the technologies not only become affordable, but they are able. But of course, we need to be able to analyze. And we are talking about the guidelines for the American College for Medical Genetics for identifying variants of concern. And a lot of work needs to be done by the bioinformaticians. I need to speak about the other side, though. The beauty of having genomic surveillance happening in Africa at the pathogen level. It's very impressive that countries have developed capacity to be able to identify which bug is causing the disease, be able to make sure that they know the strain that it is, and the African Union, of course, with the setup of the African Pathogen Genome Group, is leading quite a lot of that, and that is also relevant. So from the point of view of the content of the host, we are almost there, but not there. I think the last presenter echoed the same words. So we have the simple, rare Mendelian disorders. We are still struggling to treat those ones. Sickle cell anemia. We don't have clear. I think it's now that the gene therapies for sickle cell anemia have entered clinical trials, and we are. You can see we are trying to leap from the simple rare Mendelian defects and move to the complex disorders. And that probably needs quite a lot of work for us to be able to achieve. So, the hot areas, cancer genomics, of course now we have Parkinson's. I've talked about breast cancer, how in the past we were just looking at the histology, but it's beyond the histology. You need to be able to look at the markers. And then, of course, the issue of choosing the drugs. And a lot of this we've really gone through how to address rare Mendelian disorders. So there are several programs trying to understand. We have this program running, the undiagnosed disease. It's interesting to learn that there are diseases which are not yet diagnosed today, whose phenotypes is not linked to a clear underlying pathology. And so this program focuses on that, and the idea is to really make it possible for many of these diseases to be diagnosed. And then, of course, prenatal screening. How do we apply prenatal screening for PD? Would you want to know from whether your children will get PD or not? Those are questions to really ask. Yeah? Newborn program we talked about. So frankly, I think that is it. The tools, the bioinformatics tools. So figure a situation where the physicians who used to do a ward round and make decisions simply based on the clinical diagnosis have to go on and make sure that they understand the DNA makeup before they make the decision. So this is probably a senior consultant arguing with maybe a clinical pharmacist somewhere trying to decide which drug. And all these are issues to really look at. But that's it. I think I don't want to spend a lot of the time. This is what I've hidden from you because most of the presenters have presented something close to this. So I'm going to stop here. The linkages that need to be done to make it possible, a lot of decisions, accounting for individual vulnerabilities. And of course the moratorium, and I think here individual countries and individual consortia of countries, conglomerations have to make strategic decisions, policy decisions, to help make sure that this works. So I think different cohorts are being set up in the US to try to address some of these issues. That's it. Thank you.
Okay. He has given us, he has extended all the deep stimulation of the stigma. Thanks, Mr. Ake. I think we'll continue so that we can catch up on time. Serge, you have to ask him something. Please.
A comment. Okay, I'm looking around. Yes, I think I'm the oldest person in this audience.
I like this kind of question.
So I'm the oldest person. I'm not interested in sex any longer. Yes, my wife the same. I think we will go even further than what you are saying. I'm pretty sure what is going to happen with the children of my grandchildren. My grandchildren are now 10 years, 4 years, from 4 to 12 actually. They will not have sex anymore. Their children will not have sex anymore. Yes. So be prepared. What is going to happen is, according to me, that's my vision, Jules Verne, the woman will take away fibroblasts from the elbow somewhere. 1 gram, 1 milliliter. That's about 1 million or even above 1 million of cells. They are going to give it a cocktail to bring it in an embryonic stage. They are going to let the meiosis happen so that there will be haploid. Then they give the sperm cells of the boyfriend. Yes? So no sex around. Then they do the fertilization, and then they will culture about, let's say that about 10,000 cells will be successful, yes, fertilized. They're going to grow them for about 1 week. Then they are going to do, they take 1 or 2 cells, they're going to do the genomics on it. Very cheap. So 10,000 cells, 10,000 markers, and then they can predict by machine learning, artificial intelligence, whatever you like, they can tell which embryo will have the least chance of developing ovarian cancer, breast cancer, prostate cancer if it's a male, burnout, all kinds of problems, Parkinson's disease, Alzheimer's, etc. And then it's the parents, they can choose about their 10,000 embryos that are in the freezer. They can say, well, this embryo I would like to be implanted and to give birth. As a result, we will have 20 years later, there will be much less problems of Alzheimer's, you name it again, yes? And also you can choose the IQ. So you can say, well, I would like my boy or my girl, well, I hope they will not be able to choose the sex, but the boy or girls, but you can ask to have the progeny with an IQ 10 or 15 points higher than yours. Yes? So that will be the future. I will not be there anymore, but please, if we encounter each other in heaven, tell me whether I was correct or not.
Thanks, Serge. I think, okay, you're on now, Serge. Please, carry on.
Good. Now we will talk about sex. No, that's not true. I was asked, this is real. I was asked to talk about nanotechnology and nanobodies and usable approaches in PD and management options. The problem was a few, well, 10, 15 years ago, my father was diagnosed with Parkinson's disease and my mother with Alzheimer's disease. And I said, that's true, this part. What's coming now next is not true. I said, I leave Parkinson's disease to the Kenyans, and I will go for the Alzheimer. So I will have, be careful, it's going to be a tsunami of slides. I have 40 slides, 36 will be about camel antibodies, nanobodies, and 4 about Alzheimer's disease. So be prepared. Okay, first, a very brief introductions about antibodies. Antibodies, as you know, they are the therapeutic molecules in our body. If you are ill, you generate antibodies, those are therapeutic molecules. We can also use those antibodies for ourselves and to treat somebody else as a therapeutic treatment. We can also use the antibodies in diagnosis. We can use them to capture the antigens and to capture the molecules we would like to investigate, and we can also use the antibodies to detect whatever has been captured. It's kind of a sandwich ELISA. So the reason why we are so keen on antibodies is because they can be raised against any target, call it an antigen. They are highly specifically recognizing this antigen in one single spot, the epitope as it is called. They associate with a very high affinity, well, we can select antibodies that will bind with a very high affinity, and they can be obtained in a monoclonal form. And there is also something else. Antibodies have an extremely well-conserved architecture. It is always composed of 2 antibodies, that's what you learn in your textbooks, 2 polypeptide chains, one composed of 2 domains, a variable domain at the N-terminal end and a constant domain at the C-terminal end, and the heavy chain, the second chain, is composed also of a variable domain and 3 to 4 constant or conserved domains. And they are going to assemble in the following way. so as to generate, and they are kept together here with the blue lines, which represents disulfide bonds. Now this is a large and complex molecule. You're going to bind the antigen here at the paired VH and VL, and also there. So it's a bivalent structure, and here the paired CH2, CH3 domains, that's what we call the FC, and that's going to communicate with your immune system to tell, to give the instructions of how to degrade and how to remove the antigen that was captured. That's how our antibodies are working. But as I said, they are very large and very complex, and we try to make them smaller. So one way of making them smaller is by protease digestion here in this hinge region to produce the fragment, or you can clone the encoding genes for this region and to express it in microbial systems. That's one way of doing it, but this is still large and the expression is not always easy. So then people cloned the VH and the VL and co-express it in bacteria or in fungi. That's what we call the variable fragment. That's the smallest antigen binding fragment that we can generate from these complex antibodies. But they have a drawback. Those VH and VLs, you see there is no blue line between them, so they are only kept together by hydrophobic interactions. This means if you're going to dilute it, if you inject it into a cathedral of a body like mine, which is about, I tell to my wife, 70 kilograms, but actually it's a little bit more. But don't tell my wife. So then it's going to be diluted from a small ampoule, from a small syringe to something of 70 liters. And so if it's diluted, it will dissociate, and it's no longer binding to the antigen. So how can you remediate this? You can remediate it by putting it together with a small linker to produce a single chain Fe. And this is the smallest intact antigen binding fragment that one can generate from antibodies. This is what you learn in textbooks. Now, I was lucky, and my professor, Rick Hamers, he worked on leishmania and also malaria, and so he tested several diagnostic tests to see whether water buffaloes in Vietnam or Thailand were infected with trypanosomes. And so he published this paper in the journal Annales de la Société Belge de Médecine Tropicale. You probably know those things, the bibliometric analysis. You have the Lancet, Cell, Nature, Science, all around 40. Yes, and well, about 5,000 times lower, you have this one. Annales de la Société Belge de Médecine Tropicale. That's where he published his seminal paper. Yes, and it was picked up by somebody from Mali. And that guy from Mali said, well, we have the same trypanosomes in our dromedaries. Those dromedaries, they can be infected also with trypanosomes, with the same trypanosome enzyme. And so does your test work also with dromedary serum? I said, we don't know. Why don't you send us blood from your dromedaries? So we received in Brussels 2 liters of blood, 1 liter of healthy blood, and 1 liter of a dromedary that was infected with trypanosomes. And so we did a test with 1 droplet of blood, and it worked, the test. So it was all done. Now we had students, they were not interested in the practicals. And so they refused to take blood from a volunteer because they might be infected with HIV or hepatitis, so they refused to do this. Therefore, the Hamers said, well, we go to the animal house, we will kill a mouse, and we take the blood from the mouse. And they said, oh no, we have biology students, we are going to protect life, we are not going to kill a poor mouse for a stupid practical. And then Hamers said, well, I do have healthy blood, blood from a healthy camel in my freezers. I will give this to you. And they did it, they used that blood to prepare on protein A, the immunoglobulins. The protein A binds on the FC region. And this is what they noticed. They had a fraction here with a molecular weight of 160,000. If you reduce the disulfide bonds, you have a heavy chain and a light chain of 50 and 25,000. And so that's the normal picture I showed you before. But then in addition, they had two fractions from the blood serum of the dromedary that had a molecular weight of 90,000. And if you reduce it, there was no light chain, and also the heavy chain is slightly smaller. And so Hammer said, well, probably what might have happened is that in the camel they have these antibodies, but they have also another class here that lacks this first constant domain. This is the anchorage point of the CL, the constant domain of the light chain. So if this red domain is absent, the light chain can no longer bind, and you're going to have this type of an antibody, which we call a heavy chain antibody. We tested this, and so we had cDNA sequences around, and this turned to be correct. And also that VH domain, I should go back perhaps. So we call this the VH, and here we call it VHH, which stands for variable domain of a heavy chain antibody. So HH, good. So was this different? Yes, this is different. The variability is not everywhere in the sequence. It is localized in 3 hotspots. We call them the hypervariable regions or the complementary determining region, because in the folded domain, they're grouping together, they're clustering together to form one single platform with which they can recognize the antigen. So, oops, sorry. So here you have these 3 CDRs that are clustering together, and this framework 2 region is hydrophobic amino acid because that's where the light chain, the VL, should attach to it. It has the same composition actually, the same architecture. Now the VHH also has 3 hypervariable loops, but they are a little bit longer. The CDR1 and CDR3 at least is a little bit longer, and therefore it's very often connected by an extra disulfide bond. Not always, but regularly, in dromedaries at least. And also the framework 2, while here you have hydrophobic amino acids to connect, well, to pair with the VL, here this guy has to work on itself, so it's going to substitute those hydrophobic amino acids by hydrophilic amino acids, so that the domain, that the VHH, is soluble in an aqueous environment. Good. Where does those heavy chain antibodies and classical antibodies come from? Well, the genome of the camel has several mini genes. They have a pool of variable genes, VH and VHH, germinal genes. They have a pool of T genes, diversity genes, a pool of joining genes, and then a pool of constant genes. And what happens in the cell is that you have a recombination between 1J, 1D, and 1V to produce the variable domain. And this is going to be expressed, and also the light chain will rearrange with a V and a J gene to produce this light chain, and this is going to produce our classical antibody. As I told you, you can then clone only these parts and this part to produce the FAP, or you can produce only the variable regions to produce the single chain ID. But in the genome, we also have VHH germlines. And if that one is going to rearrange with the same set of D or J genes, then you have a VHH domain linked and is going to be associated with another constant domain that lacks the CH1 exon. The DNA is there, the CH1, but it's spliced out during the maturation of the messenger RNA. And then you produce immediately the heavy chain antibody. And as you can imagine, if we now clone only this part, then we produce this molecule, which we call VHH or nanobody, because the dimensions are in the nanometer size range. So we call it nanobody. I told this 3 months ago, 4 months ago as well. That's a very good strategy. Whenever you discover something, give it a new name if you want to publish it. Not only for patent reasons, but also if you're going to submit it after you apply for the patent, if you're going to submit it to an editor to publish, then that editor is going to say, well, nanobody, what the hell is this? And he's going to send your manuscript to reviewers. So always try to have a new name, a fancy name in the title of your paper. That's a very good trick. So which animals do have those heavy chain antibodies? Well, apparently it's the dromedary and the camel. This is the camel. The llamas and the begonias and the alpaca, they have it. So it's the only survivors in the Camelidae. Belongs to the Chilopoda, belongs to the Artiodactyla. Other Artiodactyla like Swiftworms or Ruminantia, pigs, Hippopotamidae, giraffes, goats, sheep, and whatever. All the ones in green, they do not have those heavy chain antibodies. So it is something that emerged after Chilopoda splitted from Swiftworms and Ruminantia. Yes? Why I'm telling this? Well, I want to show to you that those heavy chain antibodies and those nanobodies that we generate from it, it's something quite similar to what is happening in your body. Yes, and we understand it very well what is happening there and how they are affinity matured, etc., etc. Good. Now we developed a technology whereby we are going to create an antibody library. We have several possibilities there, and I will focus on the immune library because it's the best and the easiest. Then we have several selection methods, and I'm going to select, well, to focus on the phage display because it's also, this is professor proof. Even I, as a professor, I can do this. Yes, the others, that's for postdocs. That's not for a professor anymore. Yeast display and ribosome display, you need to be qualified. But phage display is not need qualifications. Everyone can do it. You have to be, oh, well, to have a failure with phage display, it's not bad luck. It's you wanted to fail. Yes, it's your own fault. Then we have to characterize them, and I'm going to overview the characterizations which we do, and then we go for the applications. Okay, good. So the first thing, and this was my job, that was a very demanding job. I was taking the antigens from colleagues, and then I went to Tunisia, to Kenya, to Peru, to Argentina, to Mongolia, to Kazakhstan, to Australia, to Dubai, to India, where they have camels or dromedaries or alpacas. And then I was doing the immunization of that animal. That means I give the antigen to the vets, and they are going to inject it for 2 months. And so I was waiting there until the blood was, well, the antibodies were affinity matured, and then we take about 50 milliliters of blood, and then I took the plane back to Brussels. So I had every year, a few times, a very good, demanding job. Good. So that's what I want to say is that you can find those animals everywhere in the world. Yes. So it's not, they're not only in Belgium. Actually, they are not in Belgium, but anyway. So now comes the trick. So what we do is we start with the blood from the immunized llama or dromedary or also transgenic mouse are available. You take about, well, a few milliliters of blood, here 1 milliliter of course. You prepare the lymphocytes and the white blood cells, you extract the total RNA, and you do make cDNA, standard procedure. Then we amplify, we have primers to amplify only the VHH genes. Yes? And we clone them in a specially designed vector, our phage display vector, and we transform it in coli cells. And we make a library of about 10 million clones. 1 million, 10 million clones. Okay, this is scraped from the plate, you bring it in culture, and now you're going to infect it with a phage, with a bacteriophage. And the vector is designed in such a way that the gene that was cloned, so every clone here has a different VHH gene, right? So the gene that was cloned is going to be expressed and will occur at the tip of the phage, while the gene is inside the phage particle. Yes? So you take those 10 million different phages with different VHH gene inside and different protein and corresponding protein outside, and you put it in the well of a microtiter plate that was coated with the antigen, like with an ELISA. Exactly the same. You put all those phages in there, and then you are washing away all the phages that do not bind via the exposed nanobody on the antigen. So you wash them away. The ones that are sticking, you elute them by a pH shock, You neutralize the solution, and since those are phage particles, they can be used to reinfect a fresh coli culture. And then you do this a few times, 2, 3 times. So you start on Monday, and on Thursday you put that on the third round, and you put it here on a plate. So you infect and put on a plate. You see already from the enrichment that it worked. Let it express. You pick single colonies, put them in ELISA or in culture, and the day after, the Friday, you make your ELISA, and you see which clones produce nanobodies against your target. The students like this job tremendously, because as I told you, you start on Monday, and on Thursday you know that it will be a success, because you see the enrichments. Yes? So we go to the shopping center, we buy bottles of champagne. On Thursday, we put them in the freezer, in the refrigerator. Friday, you have the results of your ELISA, and you drink the champagne. Saturday and Sunday, you're free. I had many students that wanted to do the selections. Okay, now we are going to produce them, and that's also a two-step, procedure. They have a histidine tag, you know a histidine tag, so 6 histidines, so that you can extract the nanobodies and put them over an immobilized metal affinity chromatography. So all proteins are discarded, and you elute only the nanobodies out of it. So what is the big advantage? Because I'm not that clever to invent this whole procedure. It was invented by Greg Winter and George Schmidt, that phage display, but Greg Winter, he did it for classical antibodies. So he took, for example, so after the immunization, they had the affinity matured B cells producing the antibodies. He cloned the, if you have 1 million B cells, he had to clone 1 million VH, 1 million VLs, and then to recombine it in a single chain FV, it's 1 million times 1 million, it's 10 to the 12th. 1,000 billion, yes, from which you have to retrieve the original affinity matured pair. Yes, so that's very difficult. We start with heavy chain antibodies. We have 1 million cells. We have to clone 1 million VHH, and we have everything in there. There's no scrambling anymore of the genes. Okay, this is the purification. You can see this is real samples. So this was, you extract it from the periplasm, you put it over the iMac, and then a size exclusion. This is how it looks in size exclusion. This is the salt, this is the conductivity, and the blue line, this is my protein. If you put it on gel, this is how it looks like. So it is crystal pure, ready for crystallization if you want, after 2 steps. Now something surprising. We found, and this is important for my last 4 slides. We found that, for example, we immunized with chicken lysozyme. Yes, and you have, this is the lysozyme degrading. These are the single chain FVs or the FAPs against lysozyme. You see that they are avoiding this area. This is the catalytic side of the enzyme. The catalytic side is always a cleft of an enzyme because it has to feel the substrate. And this is the superposition of nanobodies on lysozyme. They are all binding into the cleft, except for this one. So 5 out of the 6 were binding into the cleft. So the nanobodies, they prefer clefts to target. Classical antibodies, single chain FVs, FAPs, they try to avoid it. Yes? So nanobodies target other epitopes. Next thing, it's a very small fragment. We are talking about 350 base pairs, so it's very easy by PCR to reclone it and to make it tandems or 2 different nanobodies, one after the other with a space by a linker, or put GFP or whatever you like. Yes, it's very easy to engineer, to adapt to according to your needs. It's only a matter of 1 day and a half. Okay, what about the immunogenicity? Well, first of all, so this is the antigen. You see the cleft here. My nanobody binds into this cleft. This part is slightly immunogenic. the bottom part, because normally in a classical antibody it is linked to the Fc region, right? And it's not solvent exposed. Only with the nanobodies it's solvent exposed. And apparently 25% of people, of humans, have already a low amount of low affinity antibodies against this area. We can avoid the interaction of those self-antibodies, if you want, by extending here with 2 or 3 amino acids. So we put 3 alanines and dissolve the compound. So it's very easy to avoid immunogenicity risks. Okay, this is an overview of the characteristics. So the efficiency to identify binders, we are the winners with our nanobodies compared to FE or FABs. The yield, the expression yield, the stability, the solubility, we are always winning here. Yes, nanobodies and FABs, they are equally stable, but FABs are less well produced. So the specificity, affinity, they are equal. Sometimes we win and sometimes they have a better FAB with a higher affinity. The adaptability and the immunogenicity, we are also winners, and we are targeting unique epitopes. Now we come to the development of research tools. Okay. The first thing is, those nanobodies, apparently, if you mix it with the antigen, you can purify it, the complex, and crystallize it, and they are extremely good, they are kind of a chaperone. Very often proteins, they are very dynamic, and they resist crystallization, but if you fix it with an antibody, they are stable, and they start to crystallize. And so they have been, GPCRs have been, the structure has been solved in an open and a closed conformation with our nanobodies. We can also extend the nanobodies with other proteins, and then they can be used to solve the structure of very large complexes by cryo-EM. We can also do a transduction, so what we do is you take the nanobody against the gene that you selected, and you fuse it with the gene from our red fluorescent protein, put it in expression vector, and you transfect cells, and then this is going to be expressed, and you can see here where the red line, where the red fluorescence is, that was for cytokeratin, so you can see where cytokeratin is, an antibody against lamin, you can see the nuclear envelope in real life. These are living cells. And they can also start to degrade proteins with our nanobodies. We can also activate genes if a protein of interest starts to be expressed inside a cell, and you can even investigate protein-protein interactions and how to disrupt it in real time in living cells with our nanobodies. We use, for diagnostics, we use our nanobodies for ELISA. And again, because they are recognizing unique epitopes, we can select them and to generate a sandwich ELISA. So this was a sandwich ELISA, the purple ones. It's with a polyclonal rabbit antibody to capture the antigen, and then a monoclonal mouse antibody that was conjugated to alkaline phosphatase for detecting the antigen. So this is the signal that we obtained. We did it all with nanobodies, and you see on the same samples from leaflets and from wood, we had a higher signal. Yes, what kind of plant is this? This is the most important plant for retired people. This is the grapes, yes, to produce wine. If you are retired, you would like to drink wine in the evening. And so if the wine is, your vineyard is infected with viruses, then of course you have no harvest and there's no wine the year after. So I'm protecting your wine culture. With pleasure. Good. What else? We also developed the lateral flow assay. This is for if you are in remote areas and you need, like the COVID, the self-test, the very rapid test. This works very nicely with nanobodies as well. You can also design other methods. So, for example, here this was for a very difficult target. from Toxocara. It's also a parasite in dogs mainly and can also infect humans. So we have, but those worms are producing the Toxocara excretory secretory proteins. It's highly glycosylated protein, very difficult to generate antibodies against it, but we managed to have nanobodies. So we make a bivalent nanobody. We link it with a biotin, couple it to streptavidin that is coupled to magnetic beads. Then we capture the test and then the antigen, and then we have a second nanobody binding to another region that is coupled to horseradish peroxidase, and then we can do this potential stat measurement, the magnetosensor and photoamperometric assay. And we can also have a redox cycling to increase the signal even further. So you can do this in such a way that your sensitivity is about 50 to 100 times better than the classical ELISA. But of course you have to work with magnets and so on. It's a lab technology. Now, this is about nanobodies for in vivo imaging. For example, for breast cancer, we have heard that there are different types of breast cancer. In about 25 to 30%, those breast cancers are expressing HER2. And in those cases, the prognosis is bad. And so we would like to know whether the tumor is producing HER2 or not. And so what we will do is we generated nanobodies against HER2 and also other cancer markers. Because they are so small, you have a very good extravasation. So you inject it inside the bloodstream, it goes outside of the veins and it penetrates inside the tissues, yes, very rapidly, while the excess, because it's so small, the excess will be cleared by the kidneys immediately. Yes, the renal cutoff is about 50 to 60,000. We are here at 15,000, so well below the renal cutoff. So the excess is going to be cleared by the kidneys and ends in the urine. While if you work with classical antibodies, the blood stays for about 2 weeks, half-life 2 weeks in your blood, and if this is going to be labeled radioactively, then of course you're going to be exposed to a high radiotoxicity. With our nanobodies, it's within 20 minutes it's cleared from the body. And as you will see, it has the time to go to the target. So this is a mouse study with EGFR. So we had a mouse, we put a tumor expressing EGFR on one side, we put a tumor without EGFR on the other side. So there were 2 tumors here on both sides. And then you inject it with a radioactively labeled, technetium-labeled nanobody. These are the kidneys, the bladder, this is the tumor, and there you don't see anything. And it's even turning. This is magic. Good. Now, we have done a phase 1 study here in humans, of course. And so these are patients, terminal patients. They have been injected, and you see immediately, so this is after 10 minutes, this is the heart, and you can, after 10 minutes, you see already the lesion in the breast carcinoma, you can see it. And after 90 minutes, everything is gone, except at the lesions here. You have, this patient had a lesion here at the breast, and also a metastasis in the brain could be visualized, and also in the bone marrow with our nanobodies. Extraordinary. Good. I was told there was somebody from Tunisia. This is a study we have done with people from Tunisia. So this is the Androctonus australis hector, the scorpion that is living in South Tunisia, in Sfax area. And it has 2 venoms in its, 2 toxic molecules in its venom. And we generated a nanobody against each of those 2 toxins. And we coupled those in a bispecific construct. And now you do the following experiment. You take a mouse, you inject it with the venom, the total venom, subcutaneously. And if you do so, so you give it 2 little doses, 2 LD50s. If you don't do anything, after 20 to 25 minutes, all the mice will die. There's no escape. But unless you inject them intravenously, our bispecific nanobody against the 2 toxins, then all mice were rescued. Or once, after 10 minutes, all of them were rescued. 15 minutes, all of them were rescued. Even at 20 minutes, that's the last stage of the envenoming, we could still save 50% of the mice. So if you are stinged, is it stinged? But stung by a scorpion from Tunisia, and if you are a mouse, come to us, we can rescue you within 25 minutes. If you are a human, then you have bad luck. It was never tested on humans. Okay, this is something which might be of interest for you. The apheresis, this is amyloidogenic protein, the beta-2 microglobulin. So people that are overproducing it, and a label, an unstable variant of the beta-2 microglobulin, it deposits on the kidneys, and therefore you need blood transfusions or all the time, or a kidney transplant, yes? So what we do here is we have an antibody, we put on a column, that antibody against beta-2, and now you take the blood, you separate the blood from the blood cells from the plasma is going over your absorbent, and then you mix everything and put it back into the patient. So to reduce the number of beta-2 microglobulin. Sorry. And this was remember the breast cancer? If you now change the label, so this was done, what I showed was done with technetium, but if you put iodine or lutetium, then it is more energetic, it's more hydrolyzing, then you can kill the tumor. So this is the tumor. If you don't do anything in a mouse system, the tumor grows. And if you treat it with the lutetium-labeled nanobody against HER2, then the tumor is not growing and the mouse will survive without growth. Also CAR-T, you might have heard about it, and so people do not generate antibodies against their own tumors, because it's a self-antigen, so you have to help them a little bit. So we are generating this nanobody against the tumor, a transmembrane protein, and then signaling domains inside the cell. So you take from the patient, you take T cells, you transform them, you engineer them, transduction that you do, so that's to produce this CAR-T, and then you grow those cells and put it back into the patient, and these cells and those engineered CAR-T cells, they will attack the tumor. Now my last slides, Alzheimer. Alzheimer is going in the following way, as you know. So it's the APP that is cleaved by gamma secretase and base. So beta site, amyloid, precursor, protein, cleaving enzyme, abbreviated base. So we generated base enzyme and injected it into a camel and generated nanobodies. We selected the nanobodies, as you will, that's the next slide. You also notice when base is cleaving and gamma secretase is cleaving at the other end, you obtain a fragment, a peptide of 40 or 42 amino acids, and they are going to aggregate. And they are forming several kinds of aggregates. You have the protofibrils, the very early ones, and then the larger ones. But also the larger ones, they are very heterogeneous, and so you have very tiny ones, you have longer ones, and you have, well, 3 times as long. So I think it was 40 nanometers, 80 nanometers, and 120 nanometers. Okay, this is causing the toxicity and the inflammation. Now, the first thing we did was yes, this is the nanobody. Oh, sorry, this is we generated a nanobody against the A-beta, the 40 amino acids peptides, and we have one nanobody. And yes, okay. Here, what they did was they took what they did was the following thing. They took the CSF from patients, so from Alzheimer's disease patients, and also patients with a mild, mild cognitive impairment, the MCI. So when they start telling things that don't make sense anymore. And so what you see, if you take from those MCI patients, you have a very high influx of calcium. So if you take cells, you measure and you put calcium outside of the cells, and now you add the serum from the spinal fluid from those MCI patients, and you see that the calcium goes inside, so that it's toxic for the membranes, you're lysing your membranes, and that's the reason why the calcium goes in. They could measure this with high sensitivity. And now if you take those larger complexes, the ones I showed you here, this large complex, and that are also in the CSF, and you mix it here with neurons, then you will see that you get that, and immune cells, you will produce TNF, which is a measure for inflammation. So these larger ones, it's not that toxic, it's nearly neutral for toxicity, it's a little bit higher, but they are mainly generating inflammation. And now we added our nanobody to it, we call it nanobody 3, so this is the Alzheimer's disease, and you see that this is the calcium influx, and so it also lowers. And if you do it on the MCI CSF, then it's only half of it. This means that this nanobody in Alzheimer's patients, even the ones with mild symptoms, if we could deliver it, it will be less toxic for their neurons. So they will be healthy for a longer time. That's one step. Now the other step, we generated the nanobody against base 1, and we tested them whether they were inhibiting. We were testing whether they were inhibiting, and we had 3 nanobodies that were inhibiting. This one is the strongest one. Well, here it has an affinity of about 10 nanomolar, and also it has a good affinity at a low pH, at pH 4.7. Why is this important? Well, base is acting in the endosome, where you have a reduced pH. So it should bind to its target, to base the nanobody, at a low pH. So this one was binding there. So we then put that nanobody gene behind a promoter of base. So we take the same promoter as base to make sure that the nanobody will be rerouted in the cell the same way as the beta secretase, as the base one. Yes? And so, where is this? Well, we can't see it very well, but all different parts from the brain, they were all expressing our nanobody. If we put it in a associated virus that is modified to target to the brain. So it's its abbreviation is PHP/A. Yes, so that's modified, you put it in the tail, and it goes to the brain, and there it delivers the nanobody. What is the effect? You can't see it very well, but so this is, and they used a nanobody against GFP, and then you can see the plaques here, the green spots, and you can, well, from A beta, that's the green spots, and what is the white spots inside, that's the amyloids that are inside with the thioclavin that it was staked. And now if you do it with an antibody, this one against base, then you have much less amyloids, and also the surface, the area of the nanobodies of the amyloids was lower in each case. And they also measured in ELISA how much A beta 40 and A beta 42 they had. In both cases it was lower. So our nanobody can be delivered into the brain via this adeno-associated virus. There it can inactivate the base, therefore you're producing less A beta, you will produce less amyloids, yes, less toxicity, and you will live longer in a healthy way. And this is shown here. So these are the retained sociability and recognition of the memory. So you have a mouse, you put it in chambers here, in rooms, and then you treat the mouse and you put it, and so you let it, it's in habituation, then you give it a partner, but this is separated by plexiglass with holes in it so that they can smell it and they can see each other, but they cannot touch each other. And then you measure the time it takes before it goes to that room, because it prefers to stay away from it. And you see the normal mouse is here, and with our E-beta, well, nanobody or base nanobody, it also mimics it very well. And also then you have the preference for novelty. So when it's used to this partner, then you put in the other chamber a second partner, and then you measure the time it takes before it will also try and say hello to the second partner. And so if you do it with GFP, you see that it takes here it takes much longer because they know it's a foreign one, and so they prefer to stay with the one that they know before going and find the other ones. Well, if you have Alzheimer's here with GFP nanobodies, then there is no difference, and you go immediately to the other guy. Good. This is my last slide. These are the nanobodies that are in the clinic. So we have one is CAPIV. This is a bivalent humanized nanobody against von Willebrand factor to combat acquired thrombotic thrombocytopenic purpura. This was approved in 2018, and the second one was approved by FDA in 2022 in February. It's CARVYKTI. It's an BCMA CAR-T. It's against relapsed or refractory multiple myeloma. This is a CAR-T construct. And then we have the third one, envafolimab. This is an anti-PD-L1 FC, which is only approved in China. Okay, these are the several companies that we have, spin-off companies in our university. So it's Ablins that was taken off by Sanofi. Camelidase is now pre-series. That's the HER2 clinical studies that they are doing. Comfort Therapeutics, they are looking for GPCRs and try to develop drugs for GPCRs. Biotalis, it's nanobodies against viruses that are infecting fruits, strawberry, and so on. Yes. Then we have our VIP nanobody core facility. They are generating nanobodies on demand. And we have Gulliver Biomed. They are selling our nanobodies to all the ones that are prepared to pay a good price for it. And I will come to the end of my presentation. I would like to thank you for listening. And if you have any questions, you can always approach me. I will try to answer to the best of my possibility. Thank you. I'm staying around, but no sex. Huh?
You're still around. Okay, thanks very much, Serge, for that. Nanobodies. The next thing we'll go to is organoids. Eh? From nanobodies to organoids. Yes. So, I think we've had quite a day. Danny, we have, is it the students' presentations? Is that what you have? 7 a.m. I'm not sure. I think I'm seeing students' presentations and discussion, but the time. Optical illusion. I like that. We have it. Okay. Colleagues, the organizer is saying if we can start 8:30 tomorrow morning, so that the discussion we're supposed to have had now, student discussion, will start at 8:30 tomorrow morning.
The breakfast is always there. 6 o'clock, 6:30. Hmm?